So this will just be a quick recording of what I do when I want to start on a topic that I haven't learned before. And instead of just jumping right into the lectures and reading the notes in detail, there's like a lot of words here, but we'll start with these ones first as one chapter. So for the chapter of organelles, there's one part where they talk about the techniques to study cell structures. And I've scanned through the notes, not in detail, to get these keywords out that I think are important. And now my goal is not to understand each word in detail, but rather how each keyword is related to another and why each one is important. Why should my brain keep the info on this in my brain, essentially? And so you can use the notes, of course, and also resources like ChatGPT, Google, whatsoever, to explore and see how these things are important and there are similarities and differences. Of course, you cannot just take it wholesale. There must be some sort of thinking, and because you see, as for here, right, there are a few answers, and your job is actually not just to copy down. Then there will be no cognitive load on your brain, which makes it passive, and it'll be almost no different to reading a note. So you've got to prioritize which one is important, seems important to you, or seems intuitive to you. And for this, it's none of them, so I just went on to compare each term. So I just ignored this. And I was curious how electron microscopy was similar and different to light microscopy. So I asked it, and just scan through the answer. So this one, light microscopy says it uses light to see the thing, while EM uses electrons, and it seems to have a higher effect. And okay, that's the difference. Wow, rather than writing that difference right now, I'm just gonna store it in my head. So this is the 3D effect, while this is ultra, which I don't understand. So how is so it seems to be that this one is the surface and this is the internal structures. But I'll ask, just in case I mess up. Okay. Then I'm curious how, what does light microscopy see? Is it the internal structures or both? Oh shit, TM is the. Oh wait. Oh yeah. Yes. Okay, so apparently from my notes it says TEM also, although it uses electrons as instead of light, the way they detect the structures is through having darker areas that are more electron dense and lighter areas that are less electron dense. Then for the light, it's almost the same way. There's, where is it? Here, darker regions versus lighter regions or some shit. So in that sense, they are more similar. Let me read this, unless I messed up. Yeah. But this is very weird because TEM can detect the ultra structure of cells, but light cannot. So now like the consideration is, do I want to group it in terms of the way it detects the sample or cell, or do I want to base it off the function, which is what type of structure can it detect? And I think that these are two hypothetical structures that I can use, and I'm not too sure which is like the more intuitive one. They are both logical, but which one would it like stick in my mind more? And I'm not gonna write it down yet. I think I'll have a better idea once I go on to the next few keywords, which, let's go. The next keyword is cell fractionation. Okay, so it's I don't need to know like the big detail. I just need to know that okay, it's to separate the components of the cell. That's all. Okay, sure. And the next keyword is hope. Okay, these three are the steps of it. So okay, so this is more just releasing by breaking the barrier, and this is more trying to make it even. Oh wait, no, not even. To get the different types of organelles based on like the density. Yeah. Okay. Then what about resuspension? Oh, okay. So, from my understanding right now, it's homogenization is more of breaking the barrier, like cell wall, cell membrane. Centrifugation is to unmix it actually, to get the different components in like different layers based on the density. And then resuspension is to get back or like extract it out from the sample. And I might be wrong, completely wrong, but that's what I understand right now from these answers. And let's go back and compare microscopy and what's this called? Oh, cell fractionation. So how are they related? So right now microscopy is to see these cells or cell parts, organelles, under the microscope at high magnification to either see the ultrastructural details, basically the internal details, or the surface through SEM. And cell fractionation is to separate and obtain the samples of these cells, organelles. Okay. Like there's some sort of idea that you will first do cell fractionation, then you will do the microscopy. So it's obtain organelle sample, and it's like see organelle sample. That's the idea I'm hypothesizing. Once again, never just like put into stone the moment you like consider these relationships. It's just a hypothesis even after like you study it. But the more like content you take in, and then which reconfirms these structures, then keep it, I guess. But it's always changing. Like you can even change it right after like you finish studying, then you're, oh, maybe this is related to this instead, and then you like explore it and see which one is more logical and intuitive to you. And let's get back to the keywords. So isotonic buffer. Okay, so it's not to protect the organelles. Okay, it's simple. That's nice. Next one would be differential centrifugation. So obviously we had this step before, and I'm curious, what is the difference between normal centrifugation? And instead of just finding out what this term itself is in detail, by comparing it with other similar terms, you understand it more automatically rather than just going into detail. Or you also can read in detail of this with the intention to compare it with other terms. So it's increasing speeds. So I'm not an expert in this, and I want to know why on earth you need the speeds. Okay, I think they say it here, just nice. So it's more of to remove each component. Interesting. And I don't find there to be a very big difference though. That's the thing. It's just different speeds, and then you have But it's good to know this thing. It's cool, I guess. Then next up will be the last two keywords, and the question comes back: why is autoradiography important? And this keyword comes out again later on. So it's to visualize this. Okay. Then I'm curious, how is this different from microscopy? Because microscopy also uses the intention is also to see stuff. So how is autoradiography different from? Of course, the method is different. One uses the isotopes, one is using light or electron beams. Okay. So this one is more of like which organelles will uptake the radioactive isotopes. Wow. Okay. So the purpose is different. One is to observe processes, while one is to observe structures. Okay, what I'm hypothesizing right now is the purpose of cell fractionation is more separating, while microscopy and autoradiography is more of observation and analysis of the data itself. So I'm keen to just put it as like observation, and now I'm trying to think of a word to put here that's intuitive, like what detection, observation, that doesn't make sense. but I know that it's gonna end up here somewhere. That's the thing. Yeah, I could just do this actually, but nah, that's not good. So the idea being to obtain sample and then to you know what, I could just do that. I just could put obtain sample and then it's like analyze sample, and this I'm not too sure how to represent this because this goes to here, then it goes to here. But yeah, I think I can see that if yeah, I can see the link. I'm not gonna bother. So because the keywords are quite little and right now it's not much, I'm just gonna leave it like that. And to sort of anchor down the main point here before I forget. So the main difference is this is the observed structure and this is the observed processes. And if I learn any more of these type of things, I can also add it into the chart. And if it somehow doesn't fit into these two, I might have to re-chunk these things. But for now, there's only 3. And then there's just the... Oh yeah, I can use visuals too. I keep forgetting. So homogenization. The purpose of that is to break the barrier. So break barrier. And as a Valorant player, there's only one barrier. Ooh, Sage 4. If you're play, please don't add me. I don't play anymore. But so it's like breaking the. Let's just say breaking the barrier. Concept so good. Let's go. Yeah, that took longer than expected, but sure. Okay, so this, and you want to put keywords below it because I'm definitely gonna forget. So keyword anchoring. So it's breaking the barrier, and then centrifugation is mixing, yeah, mixing to separate the thing. So it's my what I'll think of is separating. I can't draw, holy shit. Ignore me for a second. 1, 2, 3. Art in a nutshell. Boom. Boom. So it's And I'm just gonna be like putting it like this because, and watch this shit. Whoa, just draw. I can't draw for jack shit. Oh no. Oh my god. Ah yes, test tube sample. Rotating. And once again, keyword anchoring, so centrifuge. Ignore my spelling. And the last one is resuspension. So what I realized resuspension is to obtain the sample. So I'm just gonna copy this over. Try not to do that unless, because if you keep repeating the same visuals, then you will mess up your memory. But for this one, I'm just gonna, I can't draw a hand. Screw it. It's to obtain the sample. So instead of just putting the keywords there, it's use visuals or analogies to sort of represent it in a way that makes sense to you or is easier to comprehend. Like imagine I forgot everything and just looked at this. I'll be like, okay, break barrier. Then to sort the stuff and then to obtain the stuff. Something like that. Okay, next up it's the microscopy shit. Yes. So how do I want to categorize these? And it's you. I could do it this way, where it would be TM and then like this is okay. I forgot what is the SEM base of. How does SEM organize? Okay, now that I think of it, this is not a very good way to chunk it, and we're just gonna remove this. I think I'll group it by function. So light is for, wait, what? I actually don't know. Shit, I just forgot about it again. I guess it's to see the full cell. This one is to see the internal. That one is to see the surface. Okay. Thank you. Holy shit. So I guess this makes it a bit easier. So it's like C wholesale, C internal, structures, TEM, and I'm just gonna copy this. Do not try use too much of the same one. It's gonna be hieroglyphics, but I'm too lazy to think of it right now. C surface.
Hello everyone, welcome to another.
And we are done. For now, I've got to go. Adios.